The overall goal of this procedure is to reconstitute the Tribolium castaneum Telomerase in vitro and to determine its activity. This is accomplished by first expressing recombinant histag Tribolium castaneum TERT or TcTERT protein in E. coli. The expressed TcTERT protein is then purified through various chromatography steps. The next step of the procedure is to culture the Tribolium castaneum beetles and to isolate the total RNA from Tribolium castaneum beetle larvae. The telomerase enzyme is then reconstituted in vitro by mixing recombinant TcTERT with the extracted total RNA. Ultimately, results can be obtained to show that in vitro telomerase reconstituted from recombinant protein and total RNA produces an active telomerase enzyme as visualized in the TRAP assay.
Though this method can provide insight into the Tribolium castaneum telomerase, it can also be applied to other systems, such as other protein nucleic acid complexes.
Generally, individuals new to this method will struggle because isolation of the beetle total RNA is extremely sensitive to RNase degradation, and therefore great care must be taken in order to ensure proper reconstitution.
This protocol begins with the expression of recombinant TcTERT with the tobacco etch virus protease cleavable N-terminal hexahistidine tag in transformed Rosetta DE3 P-lyse S cells, as described in the written protocol. Following cell harvest, sonicate the cells on ice for 2 minutes in 30-second intervals with 30-second pauses at approximately 51 watts of power. Spin down the lysed cells at 18,000 RPM for 20 minutes to separate the soluble and insoluble protein fractions and carefully remove the supernatant.